Hi guys, today we're going to be talking about cephalopods, which is a major group of mollusks, one of the major three classes that we are talking about in these mollusk videos. And so if you haven't seen the first two mollusk videos, I will link those up here for you, but go check those out because those will help with a little bit of the background of mollusks and the entire phylum's common characteristics in general. So go check those out before we move on. And if you have, then let's get started. So for some general cephalopod background information, cephalopods, like I mentioned, are one of the major groups of mollusks, along with snails and clams, or gastropods and bivalves, like we talked about in the last video. And sometimes cephalopod shells can be mistaken for looking like gastropod shells, but cephalopods are very different than snails. And we'll talk about more reasons later, but the major difference you'll see between cephalopod and gastropod shells is that cephalopods have chambers, and we'll talk about what that means later. But basically, cephalopods include things like nautiluses, cuttlefish, octopuses, squids, and the extinct ammonoids, which, as we'll see later, are super important in the fossil record. Additionally, cephalopods are exclusively marine, but can be very diverse and live in shallow, intermediate, and very deep waters. Also, some cephalopods, like octopuses, for example, are very intelligent and can solve complex puzzles and mazes, have elaborate courtship rituals, and can camouflage and communicate by changing the color or patterns in their skin. Additionally, cephalopods are large and active predators that can swim above the seafloor to catch prey and avoid their own predators. They can also avoid their predators or confuse their predators by expelling a cloud of ink, as we can see in this little Nemo gif that I show here on the left. And in terms of the first most primitive cephalopods, which evolved likely in the mid to late Cambrian period, they may have been shell-less like octopuses and most squids that we see today, but by the Ordovician, large straight-shelled nautiloids reaching as long as 10 meters in length were the first large predators to ever roam the deep sea. And this may have prompted defensive adaptations that we see in other Ordovician fauna, such as the trilobites. And by the Devonian, other predators such as fish and ammonoids had evolved to join the nautiloids in pushing those defensive adaptations in other fauna. And we know that fish are not part of the cephalopod group, but ammonoids are. Ammonoids are similar to nautiluses in a lot of ways, but are also very different for reasons we'll discuss later. But basically, when ammonoids came on the scene, they diversified and dominated so much that they became really good index fossils for the Devonian to the Permian periods, as well as the entire Mesozoic era. In fact, they are the best, most precise index fossils that can be used for the Mesozoic. And in terms of size, cephalopods, like nautiluses, ammonoids, and squids and octopuses, can range in size from less than 1 centimeter up to 18 meters or 50 feet in length. And we can see here that ammonoids got up to around 1.7 meters in size. Octopuses get up to around 10 meters in size with their tentacles stretched out. But the largest cephalopod to ever have lived actually is alive today. This is the giant squid, which gets up to 18 meters in length. And this is just based on what we know from the rare sightings that we actually get to see. So they might even get bigger than this, and we just haven't seen it. But now that we have some of the basic background information over cephalopods out of the way, let's move on to morphology of cephalopods. Here we can see that in terms of the external morphology of cephalopods, they have eyes, which are actually very well developed, and they are adapted to see very well in the dark so that they can go down to very far depths in the oceans. And they also have the tentacle sheath and their outer tentacles that come out and grab prey or are used to maneuver themselves around. Additionally, they have a hyponome, which is how they can propel themselves to swim. Basically, the nautilus can flood its mantle cavity with water and then close off all the entrances and just push out all the water through its hyponome, causing it to propel itself backwards rapidly. And this rhythmic pumping of water out the hyponome can cycle very quickly and produce rapid spurts of movement. However, this backward movement is typically only used for short-term escape, and the rest of the time they typically move pretty slowly. They also have what's called a hood, which can close over the body when the cephalopod wants to retract back into its shell. In terms of the internal morphology of cephalopods, they have chambers or camerae or camera singular, and they have septa, which divide these chambers, and they also have a siphuncle. I'm gonna go with siphuncle. And this is shown by the green line here in the middle, which is a fleshy stalk that connects all of the chambers and penetrates through a hole in the center of each septum. And this feature can control the liquid and gas movement through different chambers, and this helps to control the buoyancy and balance of the animal. Additionally, they have a protoconch, which is just the first chamber of the animal that formed. And then they also have a radula, which is similar to that of snails, which I talk about in the part 1 mollusk video if you want to check that out. Also, they have inner tentacles, and then also the hyponome again is labeled here, and then they have the hood labeled again here, and then the mantle cavity, feathery gills, the anus, cameral liquid, which again, like we talked about, is controlled by transport through the siphuncle, and the phragma cone, which is basically the entire part of the shell that includes all of the chambers except the living chamber, which is the chamber in which the animal currently lives. So the outermost chamber is the living chamber, and the rest is the phragma cone. So now that we have general morphology out of the way, we'll talk a little bit about the 6 major subclasses of cephalopods, which includes Nautiloidea, which we've already talked about some of the nautiloids, which we talked about straight-shelled ones, but they can also be planispiral or coiled in one plane, as shown here in figure D. And then we also have Endoceratoidea and Actinoceratoidea, which are both straight-shelled forms and both very large. However, they're different in that they have different cameral and siphuncular deposits. So basically, cephalopods can have calcareous deposits within their camera or chambers, and they can also have calcareous deposits within their siphuncle, and these are called cameral deposits and endosiphuncular deposits respectively. And we can see that the morphology of cameral and siphuncular deposits shown here on the endoceratoids and actinoceratoids are different, and that's how they can be told apart. Additionally, we have the subclass Bactritoidea, shown here outlined in yellow, which is another straight-shelled group, but these are very small in size, and they have a globular protoconch, which is a distinctive feature for this subclass. Additionally, we have Ammonoidea, which is also something we mentioned earlier, the ammonoids, which are major index fossils for the Paleozoic and Mesozoic eras, and we'll talk a little bit more about their specific morphology in the next couple slides. And then lastly, we have Coleoidea, which includes belemnites, which can also act as really good index fossils during the Mesozoic era as well. So in terms of shell morphology, we already talked about the major kind of anatomical features that cephalopods share in general, but now I want to talk about different ways in which cephalopod shells can be coiled or can be straight or can be slightly curved, etc. And so let's get into that. First, we have subclass Nautiloidea, which can have a variety of forms such as straight-shelled forms, which are called orthoconic, and slightly curved forms, which are called certoconic. I couldn't find a pronunciation online, so we're going to go with certoconic. And both straight or orthoconic and slightly curved certoconic shell morphologies can be classified in 2 different ways, and these include longicones and brevicones, and this just depends on basically the width of the shell. The brevicones are a little bit wider, as we can see. And then there's also coiled nautiloids, such as litudocones, which are only coiled a little bit and then straight throughout the rest of the shell, and involute planispiral forms of nautiloids, which are common forms today. And as we'll see later, the ammonoids share similarities with this planispiral type of shell morphology. Additionally, we have Endoceratoidea, Actinoceratoidea, and Bactritoidea, all sharing the orthoconic or straight shell morphology. Then we get to Ammonoidea, which has a diverse variety of planispiral and trochospiral shell morphologies, and we'll talk more about these types of shell morphologies in a later slide. So don't worry about those terms yet. We'll talk about those and identify and define those later on. Lastly, we have Coleoidea, which is also another orthoconic group. So now we can get to sutures. Sutures are a really important distinctive feature on ammonoids. Ammonoid shells have chambers like all cephalopods, but the chambers on ammonoid shells are joined together by intricate sutures, and these sutures can be different in their pattern. But before we we get to the main types of suture patterns, we need to talk a little bit about the terminology that comes along with identifying suture patterns on ammonoid shells. Here we can see a figure showing the internal structure of an ammonoid shell, showing the suture pattern on the shell in the top of the figure, and then at the bottom of the figure it's laid out so we can see the suture and each one of its saddles and lobes respectively. The saddles are basically the features that point toward the head, and the lobes are the features in the suture that point away from the head. So if we label this as we go from the center to the outer parts of the shell, we have the ventral saddle in the center of the shell, which is pointing toward the head. Then we have the first lateral lobe pointing away from the head. Then we have the first lateral saddle pointing toward the head again. Then the second lateral lobe, then the second lateral saddle, and so on until you get to the umbilical lobe, which is the last lobe of the structure as that gets to the center of the shell, or the umbilicus of the shell. So now let's look at some different types of suture patterns in ammonoids throughout time. First, we have a nautilus shown at the bottom left of this slide, which has its simple suture pattern as well as its medial siphuncle, which is another difference between nautiluses and ammonoids, is that it has a medial siphuncle that penetrates through the center of each of its septa. However, ammonoid shells typically have a more ventral or outer siphuncle that goes through the outer parts of each of its septa. And we can see this by the orange line showing where the siphuncle would be in each shell. Now, if we look at the nautilus sutures, we can see they're very simple, kind of straight, a little bit of curve in there, but nothing crazy in the nautiloid shells. However, as we move to the three ammonoid shells, we see that the suture patterns become more complex. As we can see, the first step toward this complexity is in the goniatitic suture patterns. These goniatitic suture patterns were most common from the Devonian to the Permian period in ammonoid shells. Then as we move to the late Permian to Triassic, the most common suture pattern is a little bit more complex than goniatitic, and that is the ceratitic suture pattern, which has crenulated lobes, as we can see in the picture. Then we move to the ammonitic suture patterns, which are just really crazy, kind of dendritic almost suture patterns that were most dominant during the Jurassic and Cretaceous, at the peak of dominance and complexity for the ammonoids, and this is when we see the most complex suture patterns. This evolutionary trend in suture patterns in ammonoid shells is not only really aesthetically pleasing in the fossils that we can collect and then make into jewelry and stuff, but it's also really helpful for biostratigraphy throughout the Devonian to the late Mesozoic. We have all of these really intricate designs in the sutures of these ammonites that evolved rapidly with enough steps that we can tell exactly where we are depending on the suture patterns in the ammonoid shells. Now let's talk a little bit about cephalopod shell morphological evolution. So like we already talked about, most of the subclasses do have orthoconic or straight shells, such as the Endoceratoids, the Actinoceratoids, and a lot of the nautiloids, which also include slightly curved or certoconic shells. And Bactritoids, which are actually not shown on this figure, also have orthoconic or straight shells, and this group is thought to be the link between nautiloids and ammonoids. However, once we get to later nautiloids and ammonoids, we do see that shells begin to coil more than orthoconic and certoconic forms. And within these coils, we have both trochospiral and planispiral forms. So why did cephalopods start to coil? Well, primitive upright forms such as figure A in this image couldn't swim very fast and could easily be knocked off balance based on how their shell was shaped and how they floated. However, later, to counterbalance the shell and allow cephalopods to swim faster, and actually instead of having the upright orientation, have a sideways orientation so they can swim faster, cephalopods began to coil in a multitude of ways as they diversified. And some ways were pretty funky. And these funky shell morphologies of cephalopods are called heteromorphs. Heteromorph shell morphologies, as shown in this figure, are all different morphologies of ammonoids which went through a series of radiations through the Paleozoic and Mesozoic eras. And as we can see, these shell morphologies include haematocones, which are shells that form two or more straight shafts. Then we have encyclocones, which had open or closed planispiral or helical early whorls that were followed by a hook at their end. And then we had torticones, which had helical whorls, and then some loosely formed torticones, which are shown here by the genus Hyphanthoceras. Then we had vemicones, which were irregular worm-like shells. And finally, we had some planispiral forms, which were more run-of-the-mill for ammonites. And we'll talk about the different forms of planispiral shells here. So So here we see different planispiral morphologies for ammonites, and we can see these include gyrocones, which are shells that form at least one volution, but their coils don't necessarily touch. Then we had tarphicones, in which their coils did touch, but just barely. And then we had serpenticones, which still had subcircular coils, but they were compressed more by touching each other more heavily, but they weren't quite involute yet. They were still evolute, meaning their inner coils still showed they weren't covered up by the subsequent coils. However, then we have platycones, oxycones, and discocones. We have discocones at the top here with involute shells that have oval whorls. So we can see those inner coils don't show very well because they're involute. The subsequent coils are covering them up. Then we have oxycones with subtriangular compressed whorls. As we can see, this one's more compressed and looks like it would be a little bit more streamlined and a faster swimmer. Then we have platycones or platycones, which are moderately involute shells. So they have some of their inner coils shown, but some are covered up. And these have subrectangular compressed whorls. So they were also likely pretty streamlined. Then we have spherocones at the top right here, which are subglobular and are very thick and probably wouldn't be the fastest swimmers. And they also have a very small umbilicus. And then catacones are also subglobular with an open angular umbilicus. And then planorbicones are the last shell type we see at the bottom right here. And these are evolute, so their inner coils are shown and not covered up. And these have relatively few subcircular coils. So they're similar to the serpenticones, but they have less coils. So now let's talk about cephalopod evolution. Cephalopod evolution can be shown here by this chart. We'll start at the bottom in the Cambrian and go all the way up to the Quaternary, or now, as we move through the different evolutionary events in this time scale. First, we have the earliest cephalopods, or Plectronoceras, evolve, and Plectronoceras led to the evolution of straight-shelled cephalopods during the Ordovician radiation, shown by the diversification of Endoceratoidea and Actinoceratoidea, as well as Nautiloidea, which, as we see, ends up surviving until the present. And then we have Endoceratoidea go extinct during the Silurian. Then we have Bactritoidea and ammonoids, or Ammonoidea, evolve during the Devonian, as shown here in yellow with Bactritoidea and here in orange with the Ammonoidea. Then we have Actinoceratoidea go extinct during the Carboniferous period. Then we have cephalopods decline at the end Permian due to the Permo-Triassic extinction. Then a subsequent radiation of ceratitic ammonoids during the Triassic period, but then during the late Triassic, during the in-Triassic extinctions, they had the ceratitic ammonoids declined just before the Jurassic Cretaceous radiation of ammonitic ammonoids, so ammonoids with ammonitic suture types, and then belemnite radiation during the Jurassic Cretaceous as well, making ammonitic ammonoids and belemnites very good index fossils for the Mesozoic. And we can see that belemnites in green here, as well as all of the other groups of the Coleoidea subclass, did diversify in the late Mesozoic and then again in the Tertiary to lead to the squids, cuttlefish, and octopuses that we have today. Then we have ammonoid extinction question mark during the Cretaceous Tertiary mass extinction event. However, I put a question mark here because there has been a little bit of evidence that ammonoids straggled on to the early Tertiary, but this is still under debate. So I just put a question mark there so you know that there's other evidence. And then after that, during the Tertiary, we do have the nautiloid radiation following ammonoid extinction, because the nautiloids were kind of their sister group that just kind of stayed in the background during the ammonoids radiation. And then after ammonoids went extinct, it was their time to shine. So they were like, hey, I'm going to come out here. I'm going to diversify. I'm going to take over. And that's what they did. They continued to diversify throughout the Cenozoic until recently, as they have started to decline now due to their shells being valuable to humans, which have caused their current decline. With that, I want to thank you guys for watching. Again, if you haven't seen the other mollusk videos, I encourage you to go watch them. They pair nicely with the information shown in this video. And if you've seen those and you want to move on to the next phylum that we have, that is the arthropods, which we'll be going over in the upcoming videos. And so I encourage you all to go check those videos out when they come out, or if they're out, go check them out now. And with that, thanks again for watching, and I can't wait to see you guys next time. Bye. I'm so proud of myself because I feel like I said all of those words right. Bactritoidea.